did you come on?
Oh, yeah, I just did part of it. I had some vertebrafuse.
Oh.
Now we have more.
I already had 8 of them done, so I did 2 more. Anyway, you know, it's kind of interesting how they do that. You familiar with that procedure?
That's why you're walking my face.
They go in through the front, and they just take everything and widen it out, go in there and straighten everything out, and use a Black & Decker drill, and they drill a couple of holes, and they put this plate in there and screw it all back together again, and shut you up, and there you go. So anyway, my voice is a little raspy because they moved that laryngeal nerve. So anyway, yes, my dear.
What was wrong with you? Like, why'd you have to get that done?
Because I've got multiple disc deterioration.
Oh,
It's
really?
genetics, bad genes. So it's a gift from your parents, you know. Nobody should have kids. That's what it amounts to. Okay, we got a boatload of stuff to do today.
The wood might be a little better off.
Boatload of stuff, but to share, show and tell real quick. Anybody got stuff? I got a bunch of stuff.
30,000-year-old virus, I think, found
Yes, go
found in
ahead.
the ice.
Yes. This was in Antarctica, as I recall. If I recall correctly, is that right? That was the one they found, was found in an underwater.
Sorry, that was just water.
Yeah. Thought you fell out of her chair.
No, it was just my water.
She was just so stunned by her.
I was so excited.
But anyway, it was,
30,000-year-old virus they found still able to attack amoeba as they thought it.
and yeah, the host cell in this particular case happens to be this giant amoeba that was just discovered too, living in this underwater lake in Antarctica.
Sounds like the beginning of a zombie movie.
Yeah, the end is near. The end is near. So that was a neat one. Anybody else got stuff? I got 2 things. And there were several studies that came out with this, but this was from the CDC site, which maybe you saw if you've been going to that. Hopefully you have been doing that. Is that what we're finding now with children who are basically the first study came out with young children 1 through 17. So. Anybody heard of this study? It got a lot of TV time last week about the fact that we're now finding large numbers of cases of illnesses, infections associated with young children who were prescribed prophylactic doses of antibiotics as kids, as young
Oh,
children.
we talked about that.
And from the CDC site it was the number of cases of Clostridium difficile infections in young children, which should never be happening. I mean, just a rarity. So the study showed that 71% of all the cases of C. diff infections identified among children aged 1 through 17 were community associated, not hospital associated, which meant that they weren't even in a hospital situation to pick that stuff up, which meant that we have changed their flora by overprescribing antibiotics for so long, or they're getting those antibiotics in a variety of other sources from the environment, and in turn are changing their profile of the flora in their colon, and setting them up for all kinds of infections, which later down the line could be bad news on the doorstep. And the other one was, this came off of actually one of my micro sites that I go to from time to time. Researchers have, let me just read this to you, have come a step closer to understanding how gonorrhea infections are transmitted. We never quite understood with gonorrhea. We know that it only affects humans. We're the only reservoir for that. But exactly how does it establish itself, that particular bacteria, Neisseria gonorrhea? And it said the bacteria responsible for gonorrhea are exposed to seminal plasma the liquid part of semen containing secretions from the male genital tract. They can easily move and start to colonize. Anyway, the whole business, what it boils down to is the fact that without these certain enzymes found in semen, that bacteria cannot invade mucous membranes. So it has to have that specific enzyme present in order to allow it to attach to cells and set up infection. So we never quite understood that before, so that's just kind of an interesting thought that came out. Nobody's got anything else?
I have a question about
Yes.
your first story.
Mhm.
Could some of that be like antibiotics found in like meat products that we eat?
It could be everything down the line. Exactly. When you consider that more than 50% of all the antibiotics that we produce in the United States goes into animal feed. Says something, doesn't it? So, and where does that go? You know, these animals don't process all of it. Their liver doesn't process all of it. So a significant amount of the antibiotics that we give them in food, whether those are chickens or cows, whatever they happen to be, ends up in the soil. They defecate it out. They belch it out. So we get exposed to it in a whole variety of ways. So, yeah, we inundated the environment with antibiotics, is really what it amounts to. Okay. So here we go. Whirlwind tour, hold on to your hats. We want to do two things today. We want to talk a little bit about nutrition and metabolism, and that's always kind of boring, so we'll just spend part of the lecture dealing with that one. And then the other part, we're going to get into genetics, and in lab today and tomorrow in genetics, one of the things we're going to be doing is what we call plasmid transfer. You're actually going to be working with E. coli and trying to change the genetics of E. coli in a mechanism that we'll talk about in lab. So it's fun. It's a hands-on lab. We've been creating lab monsters and all of that. So the first part, and I could spend a whole semester talking about metabolism, and I won't do that to you because, well, for obvious reasons. But there's some important things we need to know. So let's start off this way. What are the absolute requirements? And this applies to you as well. So while we're talking about bacteria here, basically, now we're jumping away from viruses. We know what those are. We're jumping into bacteria only, and this could be, we could make this application to protozoans or anything else that's living, that's cellular in nature. So you fall into this category. What are the absolute requirements for life? And you know what the definition of life is because in A&P1 you went through all of that crap. We talked about what is the definition of life? Well, you got to be able to reproduce, you got to be able to do this, that, so and so and so and so, all those things. Well, in order to do all of those things, what are the basic requirements? What are your basic requirements? Okay, let's go even simpler than that. Think of yourself sitting up in the morning at your breakfast table. Okay, nourishment, food, water, that stuff. And what we're getting down to basically, we start to talk about a couple of things. One are nutrients. What do we mean by nutrients? I mean, isn't that your hair looks good. It's a new you.
Thank you.
Who's gonna touch it?
Does it look great? Did everybody notice?
I know.
It looks great. So nutrients, and how do you define the term?
Vitamins, minerals.
Things that give you energy?
Maybe they do, maybe they do.
Maybe they do.
And so energy is going to play into this as well. And so let's put that up here because that's a good point. We're speaking very generally here. So go back to nutrients again. When we think of nutrients, what comes to mind?
Carbs.
Carbohydrates, lipids, proteins, nucleic acids, and what else do you see on your vitamins when you read those in the morning while you're eating your breakfast? Vitamins and minerals, right? Vitamins and minerals, which have nothing to do with energy, correct? You don't get any energy from vitamins, you don't get any energy from minerals. They're part of a metabolic process which may end up in the result of you getting energy, but they don't provide you with any energy. So we've got this, we get a need for energy, and we know we've got these biochemicals out there, and all of those biochemicals have what in common? The biochemicals have what in common? Chemically.
Carbon.
Yes. So you need a source of carbon, you need a source of energy. That's exactly why you eat breakfast, that's why you eat lunch, that's why you snack, whatever you do from time to time. You've got to have a supply of energy, and you've got to have a supply of carbon. What do we do with the carbon? One of two things. We take the carbon that we bring in and we use it for making more of us, more cells, reproducing, et cetera, et cetera, et cetera. Or we're going to use some of it in order to be able to build cells to, and what is that process called? Anabolism, right? Remember the word anabolism, as opposed to tearing things down, which is catabolism. Both of those together are what we call metabolism. Is that in order to build things, to anabolize, to build up, it requires, what are you going to work with? You know, carbohydrates, or whatever it happens to be, and the energy that's going to allow us to put those together. When we catabolize, what we're doing is very important, and we say, well, that's degradatory, that we're breaking things down all the time. Well, yeah, we are, but what's the ultimate goal? Is that we're going to break things down in order to get the energy back out of this again, so that we can actually do this process. So what we're trying to do is really what all the time? We're channeling energy along. It's a constant process. And God bless you. What do we find is that who tends, in the real world of the organisms that are out there, who tends not to survive? Humans are an exception to the case.
The weakling.
They're weaklings because they do what?
Inefficient.
They are inefficient in terms of handling energy. Why do we die when we get old?
Because the body stops getting the energy.
The body no longer is capable of processing all those products to get the energy back out to make new cells to keep the body going. The process simply slows down. And finally, what happens is that the catabolism can't keep up with the anabolism, and the whole system fails. That's death. That's really what it amounts to. You know, it can be sped up by disease, correct? But in the aging, the senescence process, it simply is a process of not being able to get enough energy to keep anabolizing. And you fail. Systems fail. And what usually fails first? Liver or kidneys. Exactly right. They're just key to that, and you remember all of that from A and P. Okay, so let's talk a little bit briefly about this process here and what goes on. And I don't want to go into it in a whole lot of depth so that you know you walk out of here scared to death and saying, I'm never coming back to this class again. But let's just talk very briefly about the catabolic process. How do we get the energy back out? Well, let's think back real quickly here to A&P1 lecture number 3. Coming back, okay? In which you talked about biochemicals. It all comes back, right? Okay, bad memory. No. We know that, and let's just deal with carbohydrates. Let's just start with carbohydrates. And in fact, we may just keep it as simple as that right now, knowing that there are other ways of kind of dealing with this. But in our body, what's the carbohydrate we like to see most? Something called glucose. We call it blood sugar because it's found in such high quantities in terms of blood. But one of the things to keep in mind is that glucose is kind of a universal sugar, a monosaccharide, that not just we use, but that most organisms use. And when we first started to understand how our cells do this, how our cells can take something like glucose and get energy from it, or can convert it into a larger molecule of carbohydrate, or can convert it into another molecule, something like fat, or triglycerides, or can convert it if we add some nitrogen to it over into amino acids. How did we know all of that stuff? And how long have we known that stuff? And the answer to that is, we haven't known this very long. The actual process of glycolysis, let's throw the words out there right now. And, right away, this usually puts sleep mode into people's brains. Okay. Krebs cycle.
Fail.
Chemiosmosis. Okay with that one?
God, that's a new one.
Okay, we'll go through that one. But what about this one? ETC, electron transport chain, which has something to do with chemiosmosis. All this stuff, you know, we don't like to talk about it because we don't get it. And we don't understand it well enough, and it's boring and all of that, and yet it's critically important. Well, how long have we known this as it relates to our bodies? But the past 70 years, we've had an understanding. Most of what we've really learned in terms of the actual processes that go on, we started to really put together about 40 to 50 years ago. So this is kind of new stuff. We knew this, that was going on, but we never understood the intricacies of all of this. And we still don't understand all of it. And where we find a lot of it still coming together is when we find people with metabolic disorders. We try to make some sense out of those. So you've got a metabolic disorder, you can't process this particular. That was a hard one to get off, wasn't it?
It's crunchy and I didn't want it to go all over me.
I know, and you did a great job. But the question now is, let's use you as an example. Can we do that? Okay, so there we go. What did you just put in there? What is that?
It's a crunchy, like Nature Valley bar.
Okay, so what do you suppose is in that thing? Probably significant quantities of carbohydrate, correct?
I would assume.
Yeah, and a fair amount of sugar. Sugar probably in the form of
High fructose corn syrup.
Yeah, there's 20 grams of carbs. Sugars, 11 grams. It doesn't tell me what kind of sugar.
yeah,
It just says
high
sugars.
fructose corn sugar. Yeah.
Yeah.
Sorry, but lots of carbohydrates. So what's going on right now with you? Okay, so it started in your, and you didn't chew it very long. You swallowed it right away. Okay. So what occurred right off the bat was that this takes you back to A&P. We're going to use those examples, and then we're going to pull this back to bacteria, so just hold on to your seats here. So a lot of this is review. So you didn't spend enough time really for your salivary amylases to do their job, did you?
No.
No. And what would we have liked to see her do? Chew and chew and chew, salivate, 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 roll up that dry old thing into a bolus. Remember that? Bolus, this slimy wad. And what was going on? Amylases were enzymes that did what? Began to break down, cleave down complex carbohydrates. What the hell is a complex carbohydrate?
Not glucose.
No.
It's
You're on
a
there.
carbohydrate that isn't glucose.
Give me an example.
Like bread, it's starch.
Oh, starch. Okay, starch. So the majority of what you just ate there was some kind of starch with lots of sweet stuff to it because starch has next to no flavor. Okay. So what's happened now? What we wanted to do, if you had left it in your mouth long enough, is that we started to break this very complex repeating units of glucose. That's all starch is, correct? All coming back. Hundreds upon hundreds of individual molecules, monosaccharides of glucose that are stuck together. And what did the amylases start to do? Simply cut that into chunks. Do we have simple sugars yet? Not even close. Not even close. So what happened? How long did it spend in your esophagus?
Not very long.
That's not helpful.
I don't know.
Help her out.
Take another bite and see.
Yeah, 2 to 3 seconds. 2 to 3 seconds. 2 to 3 seconds. And down it went. It plopped into your stomach. And it's there right now. It's there right now. And what's going on in her stomach with the carbohydrates?
Hydrochloric acid is
Nothing, nothing. Because what happened with those salivary amylases when they reached the low pH of the stomach?
They knocked out.
Okay, what happened to those enzymes? They get contorted, out of shape, they lose their active site, amylase activity stops. So she is no longer digesting carbohydrates. Were there any proteins in that?
I would assume.
Assume nothing.
Yeah, there's 5 grams of protein.
There's some protein in there. Okay, so what's going on in her stomach right now? Remember all this? What occurs is that we have pepsinogen being secreted by chief cells found in the gastric pits. And what are they doing? As we speak, in her stomach right now, as we speak, this is going on. And that pepsinogen is going out. The low pH produced by hydrochloric acid secreted by the parietal cells. Is this coming? I mean, you're looking at me. Parietal cell. Is this coming back? This should be all like in your brain somewhere. Okay? So they're secreting hydrochloric acid. What does it do to pepsinogen, which is a zymogen? Is converts it into an active enzyme, which is called pepsin. What is that doing right now to the protein that she just digested? She ate. She didn't digest. She ingested.
Breaking down.
It's beginning to cleave that complex protein into smaller chunks, polypeptides. Okay. What's the ultimate goal? Within the next, by the time break's coming around, and your stomach right now is doing this. Right? Churning. It's churning. And what stimulated that? Well, we don't need to get into that, but there were all these hormones and secretions that stimulated the smooth muscle of your stomach. And so remember, because we've got what? Longitudinal, circular, and oblique muscles. So what does the stomach do? There's this. Can you feel that happen? No, and you shouldn't, because if you do, then we got a problem. Okay? So, and what does it do? It's mixing up that stuff, that bolus into chyme, so that probably by 9 o'clock right now, by 10, quarter after 10, the first thing that's going to happen into your duodenum? Right? She's going to get the first squirt. Out of your stomach. Remember, because it doesn't dump, right? If we got dumping, we got a problem. We get a couple of mLs of this digestion going on now, or digestive material going into your duodenum, which immediately triggers a whole bunch of things. And what does the pancreas start to do? Come on. It starts to secrete pancreatic enzymes. Most of these are going to take the next step with the carbohydrate, which is where we're going to follow right now. So where are we with that then? In about an hour and a half, now what we're going to finally do is break up that carbohydrate that we started in your mouth. It's now being broken up into shorter saccharide units. Finally, what happens is we start to move this along the cells that line your small intestine. What kind of tissues are those? Histology-wise, simple columnar epithelium with microvilli. And what are they secreting? Brush border enzymes whose function it is to finally do what? Cleave that complex carbohydrate down into monosaccharides. That's the whole story from lecture whatever that was in A&P2. She'll all come back. You got the whole thing in 20 minutes. Okay? So what happens then? Absorption, correct? Absorption. And the absorption is not quite so easy because remember, how does glucose get absorbed across those simple columnar epithelial cells?
Is that active?
It's co-transport with what? Sodium. Exactly right. You named it. So what happens then? Now we're finding, finally what happens is that you're down to monosaccharides. Most of those are going to be what? Glucose. Are there going to be other monosaccharides in there? Absolutely. Some of those are going to be things like what? Talk to me. Come on.
Galactose.
Okay, fructose, galactose, etc. Mostly fructose,
Maltose.
could be some. So what does your body want to do with those? It's going to use those for one of two things. Now here's where we get back into the story again. It took us a while, but we used you as an example. What we're going to do is take that carbon-based compound, which happens to have what with it? Hydrogen and oxygen. And in turn, we're going to do one of two things. One of which is to say, I'm going to send this glucose to every cell in the body, and every cell is going to get some of this stuff, and they're going to use it for what reason?
Energy production.
Catabolism, to produce energy to get anabolism done. Or some of it's going to end up in your liver. And what happens in your liver? Excess glucose. I know it's Monday morning. I know. Excess glucose, glycogen. So the liver says what? Excess sugar, we can't afford to lose it. Don't urinate it out, because if that shows up in your urine, we got a problem. Right? Okay? So it says, I'm just going to store this extra stuff in the form of glycogen, and I'm going to store it in what cells in your liver?
Hepatocytes.
Hepatocytes. Hepatocytes, exactly right. So let's focus right now on what happens when we catabolize. How do we get the energy out of it? And all we're going to focus on here is going to be the sugar. So how do we get the energy back out? Well, maybe, gosh, the story gets wilder and wilder. Where did the energy come from initially?
At some point, it's photosynthesized by plants.
Okay, so let's go back to 3rd grade. Can we do that? Yeah.
A time machine?
Pretty good tree.
Little hole there for a squirrel. Okay, a tree. It is a nice tree.
What kind of tree?
Oh, with an owl in it.
Genius
It's a
vine.
squirrel. Oh, it's a squirrel.
No, there's your owl.
Bird in the tree. This is about the way you learned it.
Right.
Okay? And remember how some of you got to be sons? And you got to dance around the class? Some of you got to be trees? You stood in the middle and didn't move. Remember this? It's all coming back. This is 3rd grade stuff.
My class wasn't that fun.
Yeah, curses. Well, let's make up for it. So we had energy being released, and the energy that was released was picked up by pigments, this green stuff in the leaves of this tree, and the ultimate goal was to do what? Do something with that energy. We called the process photosynthesis, which you may have even heard when you were in the 3rd grade. Photo means light, synthesis means to make something. So we're going to use light to make something. Well, our ultimate goal over here is this. And the question is, where are those things? And so we've got a bird up in the tree who's doing what?
Breathing.
Singing it. At the same time it sings, it's
Letting out smoke.
exhaling. And what is it exhaling? Carbon dioxide. So here comes the carbon dioxide. And in turn, what does the bird do from time to time? Usually on the windshield of your car. Yes. But one thing about birds is that what? They mix urine and feces together. That's why you get, they've got chunks in it as opposed to just aqueous. Okay? So here comes droplets of, let's just consider the aqueous part, and there's also going to be rain that falls. Here comes the water, in comes the CO2, the plant has the unique capability of being able to take that energy from the sun and link these two inorganic molecules together to form an organic molecule, which is sugar. So now, all the energy, when you think about that, all the energy that allows you to get up in the morning, to be here, to pick up a pencil, to do whatever you're doing, to think a little bit, I need to think, I'm trying to follow what he's saying, it doesn't make any sense to me, so I just won't pay any attention. But even that involves energy out of all of this stuff. What's going on with all of that is that all of that energy that allows you to do all of that originally came from where? The sun. The sun. Are there some minor exceptions? Yes, there are, but we're not going to talk about those right now. So the majority of it came from there. So what we're trying to do then is get the energy back out. We want to get the energy out for the reasons we talked about already. Notice what she's doing now.
You drove her to drink.
Drove her to drink. She's drinking water. Why would she be drinking water? Don't say because she's thirsty.
It's necessary for the metabolic
What?
process.
What happened when you took molecules of glucose and linked them together to form a complex carbohydrate? What was the process involved? Again, this is dehydration synthesis. Dehydration synthesis, which meant that when you took, don't yawn, when you take one molecule of, in this particular case, glucose, but it could be an amino acid, to link to amino acid, you link it with the next one. In order to link these two together, what was lost? Water. Dehydration synthesis. Why is she thirsty? Why did her thirst center kick in? Because now, in order to break these apart again, she has to put water back in. You are the epitome of an example for us today. I couldn't have asked for a better one.
Well, you're welcome.
I'm glad you're here. Okay, so how do we get the energy back out? Well, in your cells, tell me what you know. In simplest words, how do we get the energy back out? And what do we want to make? We don't want to lose the energy, right? Because the energy that's lost is gone forever. It goes out into space and it's gone forever. And some of it's always lost. So there's always some energy that we try to get out of these molecules like glucose. We saw how we made it. We'd like to get this energy back out to get stuff done. What do we want to do? We want to minimize energy loss out of the process. In our case, though, we like to lose a little bit, but what do we do with it? We store it underneath fat pads, and it's called maintaining body temperature.
Regulation.
Yeah. So that's an important. That was a major yawn. That was a major yawn. So that's an important
That's another part of
reason for.
metabolic process too.
No, that's
we're not there
that's
yet.
a biological behavior that is just. I don't know where that comes from.
You need oxygen.
But yawning doesn't seem to increase oxygen content, so why do we do it?
Is it just like something weird with our brains?
Yeah, it is. It's weird. So you just did something weird with your brain. Okay, so let's get back on subject here. So how do we get the energy out without losing most of it? What we want to do is store it again, right? So we're going to take this, let's go to our glucose, C6H12, and come on, work with me just a little bit here. Just try to get your interest going so we can hang in there with this. We want to break it apart because we want to get energy from that to get work done in the cell, which we call metabolism. So how do we break this apart? And what are we going to do is reverse this process, correct? Because the end result of this is going to end up with what?
Water.
And?
Carbon dioxide.
Okay, so exhale, which you just did, major one. Most of what you just exhaled is carbon dioxide from the breakdown of glucose. Okay, so how do we minimize that? Well, we want to store the energy as best we can, and how do we store it? No. Well, you've got a point there. I'm looking for, and you're absolutely right.
ATP.
Yes, we're going to store it in a molecule, and the most important one, not the only one, but the one we're most familiar with is something called ATP. Tell me real quickly what you mean by it. And I know, again, as soon as you hear these words, your interest level goes off the table.
Adenosine triphosphate.
Okay? Anybody want to help me out? You're darn close. The A stands for adenosine. What, that sounds like what? Adenine, and you would expect to find adenine where? In DNA or RNA. Okay? So is there a relationship there? And obviously the answer is yes, but we're not talking about that now. So how do we get to triphosphate? Because tri means great. So we got this,
Diphosphate.
and we're going to add phosphates on it, mono, di. Third one, the squiggle means what? High energy. We're trying to create this molecule called adenosine triphosphate. High energy holding molecule. Again, what's the purpose for all of this? We don't want to lose the energy as we break apart this molecule of glucose. We want to save as much as we can. Some always gets lost in the form of heat. Heat's okay, but some of it just evaporates off and goes away forever. So how do we do this? Well, we didn't understand in human cells for a long time. We knew that we had parts of this all put together. And really in the 1920s and the 1930s is when it starts to come together. And where we add more detail to it is really in the 50s and 60s. This is not long ago. But the first step involved with this was something called what? Glucose splitting. Common name for that?
Glycolysis.
Sure. Okay, I'm not asking you to memorize all the steps in glycolysis. Not asking you to memorize all the enzymes. But what is important for you to note, a few things are these. The ultimate goal is to do what?
Split glucose?
Take this, right on, is to take this molecule and split it. And to keep it even simpler out of all of this, what we're going to do is follow the carbons. With me? So the word glycolysis means glucose splitting. So we're going to take this 6-carbon molecule and break it down into what? 2, 3 carbon molecules. Let's keep it real simple. Now, in order to do that though, because glucose kind of sits there, it's not an active molecule, what do you suppose we would have to do? Energize it somehow, right? It just sits there, it doesn't do anything. So we're going to have to energize this, and we're going to do that by adding phosphates. This process is called, which you're going to see over and over again, phosphorylation or phosphorylation. Either one's acceptable, which means that we're going to add that high energy phosphate. Where would that come from? ATP. It's simply going to do what? Donate some of its phosphate. So what we're going to do then is add energy from ATP. And we're going to do that twice. End result of this is that what we're finally going to do is break apart this molecule into 2 3-carbon molecules. At the same time, we're going to get energy back out. And what are we going to do with it? Phosphorylate a couple of ADPs into ATP. What I want for you to know is this. What did we do to split the glucose? we had to burn some energy. We had to spend some ATP. How many did we spend? 2. We actually got 4 back, so we made some, even though we had to spend some. Pretty good investment. Next question is, where does this happen? Well, when we first studied this in bacterial cells, we realized that this simply happens in the cytoplasm. All the enzymes that are needed for all this to occur, and there's a boatload of them, all those enzymes are simply present in the cytoplasm. So what would we do next? Make the application to humans, to our cells, and what we found in the 1950s and in the 60s was the same thing. Where does this occur in our cells? In the cytoplasm. So all the enzymes are simply found right there in the cytoplasm for this to occur. One other thing happens here of note, and that is that one other thing gets released at this point. That in the process of this, and what is this? Is this anabolism or catabolism? It's a catabolic process, it's catabolism. That one other thing that occurs out of this process is that this gets released. Hydrogen. But what's the problem with hydrogen? pH. Yeah, pH problem. Because hydrogen ions do what to your pH? Drops it and becomes more acidic. So in this process, now we've started to make some energy, get the energy back out, and where did the energy come from? You got it. But we also had some hydrogens released, and we can't leave them go. So the point of it all is, somebody's got to pick up the hydrogen. Somebody's got to pick it up, because we can't leave it floating around. The cell goes into acidosis. What happens? Enzyme systems fail, the cell dies. So somebody's got to pick it up. This is like a babysitter. So you've got this child, or these 2 children, and you want somebody to come in and take care of them for a while, but you realize these are hellish children. Okay?
Thing 2.
Which, I'm sorry, what?
Thing 1 and thing 2.
Yeah, worse than that. Think worse than that. So someone's going to come in and say, I'll watch them for a while, but eventually, you better come back and get them. Because if you don't, I'm going to kill them. So the question is, who's going to pick up the hydrogen? And we've got something called a hydrogen acceptor. A hydrogen acceptor. And there are loads of those in the cells. Loads of them. Which is the one we're dealing with here? Anybody know? And I'm not going to ask you to know what the word means, but this is it. NAD. Nicotinamide adenine dinucleotide. Just remember NAD. NAD does what? Accepts free hydrogen. For, not forever, but for a period of time. Okay, so we've gone this far. We've made some energy, but we still have 3 carbon molecule, and with that again, there's still hydrogen, right? And oxygen. I'm just asking you not to know that. You don't have to remember those numbers. What is another name for this molecule? This 3 carbon molecule goes by a couple of names. What might you, if you had Dr. Singh for chemistry, this should roll right off your tongue. I'd be so disappointed if somebody doesn't say it. I'll get it for you.
Hey, Lottie.
Did you show?
Leanna says pyruvic acid.
Damn straight. Did she really say
She
that?
really said it, she wouldn't say it out loud.
That's sad.
That was well done. I'm so proud of you. Pyruvic acid, or what's another name? Pyruvate. Same stuff. Okay. Now, that becomes an important molecule, and we'll come back to it. But let's just finish, let's finish the story here and we'll take a break. So this is all going on in the cytoplasm. Still, boatload of energy found in this pyruvic stuff. In our cells, typically we have, well, I shouldn't say that. There is a most common way of dealing with trying to get rid of the rest of this energy. And let's simply take a look at those. Because how we get the rest of the energy out of this is dependent upon one factor. And that factor is who's finally going to accept the hydrogen. So let's use three terms. These are forms of what we would call respiration. And again, what comes to mind when you hear the word respiration? We think of breathing. We think of respiratory gas exchange, correct? That's what comes to mind, yeah.
What is the last name? I'm sorry.
I'll get there, because you probably can't read that.
I did not read that.
No.
It's like
And
Greek.
so I will interpret this for you shortly. So we have three kinds. What we call aerobic respiration, which is what all of us are doing in here right now, which means that we're inhaling oxygen. And the question is, for what reason do you inhale oxygen? And if you give me the answer to live, I will throttle you. Okay, because that is not the answer to the question.
What it is.
Exactly. But what is important, though, is that what must be there? Some form of oxygen. So the respiratory gas that we need in this particular case is oxygen and aerobic respiration. Then we have an anaerobic, and an as a prefix means what? Without. Without oxygen. Which means that this is the kind of respiration, but what cannot be present? Oxygen. And then your question would be, what do you mean oxygen cannot be present? Well, we're going to see that it actually can be toxic to these cells if oxygen is there. They can't be in the presence of oxygen. So these are O2 no-nos. The third is another type of respiration, which, let's do it this way. This type of respiration is called fermentation. Fermentation. The organism that undergoes fermentation is a facultative anaerobe. So three types of respiration. Number one, aerobic. Number two, number three, fermentation.
But what was this at first you wrote?
Beat him
Why
up.
do you want me to do that to me?
No, he goes
Gosh, it's a
first.
dark blue
I
shirt.
know.
It's her orchestra shirt.
Is it off?
There's still some white spots.
Oh. I teach you to sit in front. So facultative anaerobes undergo the process of fermentation. Okay.
So why did you put facultative and anabolism or whatever first at the bottom? 'Cause I What's the difference?
This word that I wrote that you couldn't read was that,
Okay.
facultative anaerobic.
But did
So-
you have anabolism or something there?
No. It was just my spelling.
Oh, okay.
So 3 kinds of organisms. We can be categorized into one of 3 groups. We are basically what? Aerobic. Although from time to time, what can we also do? We can go into a facultatively anaerobic mode. Give me an example.
Lactic
Working
acid.
out.
Yes, exactly right. So what happens when you've got skeletal muscle that is working extra hard and what is no longer available? You can't get enough oxygen to it. It goes into a fermentation, which we call lactic acid fermentation. Okay, so we have to explain it. So now you can make some applications yourself out of all this. Okay, we'll do one thing and then we'll break. So we've got 3 options here in terms of what? How to deal with this. Let's do aerobic real quickly. You need to stand up and stretch. Is everybody
I guess
fighting?
I'll have a couple
We'll
minutes.
be okay, we got
till
to.
I-
Okay, can you go 3 minutes?
I suppose I probably could.
Okay, that means I'm going to do the whole Krebs cycle in 3 minutes. Okay.
So we go a lot faster in ourselves.
Yeah.
So I should be able to.
You're funny.
Okay, so let's go aerobic respiration real quick here, because that's what's going on in everybody in this room right now, almost all the cells in your body. Aerobic respiration, which means that somehow oxygen is going to be available. And again, what we know about what goes on in our cells is really what we learned from the bacteria, and the yeast to a certain extent as well, between the 2 of those. So what happens here? We've got this pyruvic acid, and it's got loads of energy into it. We eventually want to do what? Break it down into carbon dioxide and water. That's our ultimate goal, which is what we started with, correct? Say yes. Yes. So where do we go with this? Well, there is a party going on. The party going on is called the KC.
The Krebs cycle party.
Exactly. Okay, so it's this party, and in this party, what are there? There are a whole bunch of enzymes.
Just like a dinner party or a rave or like
This is more like, it can be anything you want to, so you can let your imagination run all over the place. The important thing is that not anybody gets invited. In fact, you can't get in unless you got a date. Okay? So what we have here is this pyruvic acid, pyruvate, would love to go to the Krebs cycle party. But, first of all, there's a problem. You have to look a certain way. And one of the things is that you need to lose a little weight. It's like that. So this 3-carbon molecule of pyruvate says, I'd really like to go, so I will lose some weight. I'll lose a carbon. And I'll lose some oxygen with it. And I'm going to give that carbon off as CO2. Exhale. There it goes. Okay? So now we're left with what? A 2-carbon molecule. Anybody remember what we call that? It's oftentimes referred to as an acetyl group. And it still can't get in the party because it needs an escort. So it goes out and hangs around at the corner and goes, hello, sailor, until finally somebody comes along and picks it up. Well, who picks it up? No, something called coenzyme. Anybody remember? Coenzyme A, exactly right. So we got coenzyme A, COE as it's sometimes referred to, which picks it up. And lo and behold, coenzyme A, which we now call acetylcoenzyme A, because they're kind of a couple, comes over to the party, and the door opens wide. You lost a carbon, you look great. You got an escort, you're looking good, come on in. What happens next? Stifled. Just when you think the evening is going to be great, the evening is going to be fun and exciting, what happens is coenzyme A finds somebody who looks better. Yeah, the story gets ugly fast. And poor acetyl group is left there alone. I'm looking for a tear. And what happens is coenzyme, excuse me, the acetyl group then simply gets bounced from date to date to date to date in here. Nobody wants her, or it could be a him. Nobody wants this person. End result being, though, that what happens? The rest of the carbon gets released. The rest of the oxygen gets released with it. And now all our carbon is gone and all our oxygen is gone. But what remains? What did we find over here? Hydrogen, which we said we were going to have a problem with. And then at the same time, we had hydrogen given off here. Where has the carbon and the oxygen gone? In the form of CO2, and it's simply, if it's a bacterial cell, it dissolves off. What happens in us? We exhale it. There it was. So what will happen in another hour is that you'll take one deep yawn and you'll blow off a whole bunch of CO2, and most of that CO2, the carbon from that, came from that thing you ate. Finish this. Say what?
So with these extra hydrogens, I'm just thinking,
Ooh, go ahead.
all right, with these extra hydrogens and stuff like that,
I'm all
they
ears.
can't just be floating around, right?
Nope.
So eventually does like oxygen pick them up and we pee?
Well, let's see if maybe that's the scenario.
Okay.
So we never have free hydrogens being given off. They always get picked up by somebody. And in our body, what happens? We have what's called what, buffering system? Remember all that? So in a sense, it's kind of the same scenario here. So the hydrogens are going to be picked up by NAD or something similar to it. I'm not going to ask you to know the difference between them. But the question then is, remember, these are problem children. And we need to get rid of them.
Wow.
So where do we go with them? Well, now we have some options. One of which is this thing called the electron transport chain, and the other is called chemiosmosis. The electron transport chain we've known about for 50 years. Chemiosmosis we've really only known about for about 25 years. And we realized that the two of them work together. In your cells, let's just deal with this real quickly. In your cells, where did this process happen? Glucose splitting, glycolysis, in the cytoplasm. Where did it happen in a bacterial cell? In the cytoplasm. Where did this process occur? The Krebs cycle, yeah. In the mitochondria of our cells, correct? In fact, more specifically, where in the mitochondria? What does that mitochondria look like? AMP1 again. That thing looks like this. And it really contains what? Two membranes. Remember that? membrane out here, and then a membrane in here. Both of them are biphospholipid membranes, but slightly different from one another. And it looks like this one was actually, what, too big to fit, and so what did somebody do? Squeezed it in there. And where are all the enzymes that allow for the Krebs cycle to occur in our mitochondria? They all lie on the inner membrane of the mitochondria. What's the problem with bacteria, and what do you know? We don't have mitochondria. There are no membrane-bound organelles found in bacteria. So where does this happen in a bacterial cell? No. The cell membrane. So it's going to occur, this is the mitochondria, here is the bacteria, which it happens to be a what? Bacillus. So all of those enzymes are embedded in the cytoplasmic membrane, the cell membrane. Now hold on to that thought, okay? And let's break now and come back, and then we'll finish the story off and we'll move on, okay? So run around for 15. I'll get some fresh coffee.
I'm not doing my test.
Do what?
I'm not doing my test.
I'm almost done. I just have to record it. I didn't
So give me extra credit.
So you haven't posted the test card?
No, they're all done, but I'll post them today.
Sorry about being late.
We'll get them out there.
Both sides?
No, it's just
Huh?
Yeah, right.
Yeah, I mean, either way, we'll have to study for a while.
Yes.
Yeah, for the summer. Yeah, I didn't do it till 9:30.
I just know.
I was seeing it. Did you see my girl was like waiting list of like
It's funny to have the
girls on my calendar.
I looked at your girlfriend's medication. It was the perfect analogy for it.
It's like a little thing like
No,
that.
let me just finish. Our nursing
I haven't got far,
one.
but
It's
you know, top up, so it's a lot.
Which is
You
next.
know what I mean? I feel like it should
This
have
was
been.
sleeping so crappy and then last
I like
night.
every
my
time
report,
on
it's
that
not.
test.
I slept
Very
very well.
well.
I'm
I'm
actually
actually
like,
like,
March 8th, 3rd, 9th. Are you banned again?
still.
No, I
How are you
read
getting it?
That's what I'm doing right now. But I'm trying to think back over it.
Because I like when it's out.
I'm one for they started. But I probably will be here.
Yeah,
It's okay.
I really like the times.
I mean, I'd rather get it when I see, but oh my God. I'm scared to give my extension to my manager.
Oh, I know. In the summer, I work at a message place, and in the summer we have Tuesday morning classes. And I'd be like, sorry, boss, can't do that shit.
I know, I get a, where you work at? You work here?
She works in Humana. I actually.
Right. You meant,
They just
I
changed
know.
our benefits.
I
They just changed our benefits to try to help. So I gotta work, I was working 16 hours a week and still to get my benefits. But now you gotta work 40 hours of a period. And I was only working like 32. Well, I was on the schedule like 32 or more.
You don't need to remain all that pre.
I don't know how to do that. I'm gonna have to work about every Friday and Saturday or something. I don't know.
So do you work for Tri-Health?
Yeah, I work every day.
Are your benefits, do your benefits start day one? Or do you- Okay. Because I've been contemplating trying to find a job here.
I know,
Because I
I
just think it's like.
just want to finish nursing one first and then I'll.
Well, I was thinking that, but like, with my work schedule, would be so much easier to just work like 312 somewhere. Versus like 213 to 10 and a 6.
Yeah, that's you still working a lot. You gonna work like that the whole time?
I have to. I don't really have much of a choice. Yeah, Thursday school if I'm not working 40 hours a week.
Because some of the stuff is.
Or I'll be homeless.
Would you work on Thursday?
But I can't, so like now I work,
That's your.
like Wednesdays and Thursdays I work 7:30 a.m. to 8 p.m. Fridays I work 1:30 to 8. Saturdays I work 8 to 6.
And then.
He's tough. So he works
But then he works.
Yeah.
So he's home with your son.
Well, we're doing daytime work, yeah. But it's
Yeah, that's how I was working. Like, the first time I had got work, this was I paid up to my lease, but like what I'm gonna do after that.
What'd you say?
Well, it's
I don't know.
I could get on like computer or something, I could like. Work on like.
That's why I said I'm gonna start trying to pick up here more, maybe go in between, because they always need help, something like that. So maybe try to do that, but I said I guess for the, it's only the, I'm like, it's only the month of July.
I know, and it's what, we get Fridays off.
Yeah, it's okay. I can go to work on Fridays. I can go to work, like, cause our first shift starts at like 4 in the morning, so I can go in and help them with rounds from like 4 to 8 and go to sleep.
Yeah.
It's only 9:30 to 1. And you know single is now.
After you have it. Yeah, I'm not even going to, I'm not sure you're going to.
If I read it.
It's just every day.
I don't think it's bad at all. I just
I
be like, oh my
see
gosh,
it'll
as
be
soon as
done
that's
by-
over, we're gonna be starting the next time.
Mhm.
Cause when the fall time starts, I just look like the 19th or something.
Yeah. It's like the third full week, or second full week.
But that's okay.
I don't have any summer classes, so maybe just.
I'm gonna take ethics next summer.
See, I'm done with all my stuff. I'm taking farm math. What day was the farm math?
I'm taking farm math
Tuesdays?
on Monday.
Next time? Monday?
Because it works the
What
rest of
time
my
is it?
personal stuff. I don't remember. I have to look at my paper.
What time was your, you were that night, didn't you?
No, mine's 9 to 10.
Okay, yeah, I need to take my classes.
Which wasn't bad last week.
But we got nursing on.
What'd you guys do last week?
Well, we did the same thing. Like, it was actually interesting stuff. So it made it look good. And Dr.
Remember what?
A project.
Nice, what's that?
It's like a 30-second a goal in our class and how we achieved it by being in that class. It's, I really don't know more of it, and I kept asking my last and I kept asking questions because he wasn't
Is it
confirming.
due tomorrow?
Oh, see if there's anything left for Friday.
Yeah, I forgot.
She just cashed it, like, 2 years ago.
But don't we got don't we got a lecture on
I
Monday?
mean, I had it in there, but you just planned it and that she was.
So it had to be like on the.
Are you talking about your payment? I turned it in so long ago and she just did it on
She
Friday.
cashed like a month after I turned it in.
Yeah.
And I'm like, thank you for that. I'm like, I didn't have enough money, I'm like, no.
Yeah.
But yeah. You said they got one on Monday night. It had to be a night then after the lecture or afternoon.
What's after the lecture?
After the lecture.
It's like.
That's what I forgot to
I don't know
do.
the difference.
That's so late. My brain is shut down.
Oh, yeah.
Maybe I can do it on the day.
I wonder if like, the sociology is Tuesday from 1:30 to 1.
however much my.
So that gives me, you know, like a minute to get to another class. Someone in the actual semester. Because I don't want to be here 40 weeks just because like.
I can hear, or I have no idea.
Yeah, well, that's.
I think mine's like Monday, Wednesday, Monday, Tuesday, Thursday.
Monday, Tuesday, Thursday. No Friday.
It smells like weird.
But I was trying to keep my schedule the same, I just can't do it, that's why I said, if I don't, I have to go out, and I was there, I didn't feel bad, because like, I had some shit, maybe mad, because they work with me on my schedule, everybody else.
No, that's what it is.
Changing the days I work, which makes their schedule change.
Right. I was like.
So I'm thinking, my medical big change, if I keep it how it is, I'm
I'm not going back.
Do you have like health insurance?
not have, I have my 401k.
Yeah, but I have to do.
That's like, I probably should have.
Both could carry it to my apartment. They're not as good for just what I have in Humana. So, I mean, it's something that we can do,
When optional, I wouldn't do. But I mean, I can still work, I know I can work, but I know, you know, what's like, what I don't work. Like, I'm yesterday, I
You want to clear
worked Thursday, tonight, but I ain't gonna work.
Find all the numbers and when it was and this one.
Do you even play anymore?
My dad was so bad.
I just didn't write down
Yeah, but it was so.
No, like, I was on the dash, but she didn't find it.
Oh, okay,
But then, and then, they tried to, and I just,
She never ended up, because I gave her all the information.
Well, but like I mean, when I bought my first one, but I mean, I bought it because I thought I was going to be that as a baby, but it's it's a lot better than what the starter one is. But you have to go in there and start, like, if you can, I don't know if you do private lessons there, I'll take it, but at the time, I asked my private lesson teacher or somebody like that, or, you know, we went to Antonio violin shop, they probably have other ones like that, but they said sometimes, like, Sam Nash and stuff don't have his degree, it's like, as compared to, you know, it might be, or some manufactured like machine like the Sam Nash, and then you'll have your personal Harvard that make your violin shop. Or you just Google, that might be, I don't know, but just like Google clarinet shop or something like that in Cincinnati, but like I said, they'll know. I mean, you know, I might be like, yeah. I said, but then, you know, you might be separate I like those type stores because they'll be able to tell you, this wouldn't hurt this one. If you want something like this, you know. I mean, sometimes they don't know as much. Like, since they have so many instruments, it's like, oh, I heard this one's good, or, you know.
That's why I was looking at I always wanted to work at a lake.
Yeah, because you've never clarinet, you don't know anything about violin, like, you know, it's like, you don't. And then they might not know that much.
It's just like, oh, farm assets.
Oh, okay, is that like kind of like a
It's like a shop like that. I know.
Okay, I go somewhere like that.
Yeah, and I always get my moves from there, and they're really
No,
good.
you go through.
I'm kind of best at it.
You just, you don't have, like, you go to Sonic and of course, it's
But you, like, log in,
I don't know.
Which is fine, like, I don't.
all up front.
I mean, they'll know where you
Down
live.
payment, the register next Thursday.
And they don't know where you live, probably like really
Are we friends now?
like, you can take it home.
I'm too far away.
Just go find it.
And I'm like, looking
I would have, I thought about it, I had, like, two personalities, I would.
Yeah, right back in. How was she at first?
I mean, you can figure it out.
oh, yeah.
They're all.
Out the car, that's the last thing.
I think you can roll this till like 12.
Yeah, I'm about to say, you could go anywhere.
But I mean, you know, it's a lifetime thing. It's not like you don't have to keep buying them every year. It's like, well, they because I'm gonna do that to trade it back, and they just say better. So I can just keep it's just, I mean, you could sell it back, but you might get like, I mean, it started. Yeah. I don't know. You might get more. I mean, I wouldn't think you get 900 for it. You just gotta pay I bought it too because I kept the camera. It costs more now.
It absolutely
It's hard.
Like I said, I can tell you that straight side of me. It's only like two of them for us. It's like the road we're in, so I don't know. But, I mean, you know, like I said, those stores like that, I go to. It's just like, I mean, you might have somebody that's even clarinet expert, but it's kind of like going to Walmart. Like, really? And then we got these questions of people that actually probably play. And since it's a lesson. Yeah, I would say yeah, that's probably
Really, it was so weird.
That was like pretty much because we kind of lived here a little bit. They can tell you like the inch size.
Yeah. Okay, I can see that this one's bigger than the other.
It was something like that. It was like one of those big, like, crazy little players. Oh, boy, I've been one of those.
Other students are
Susan phone.
my next door neighbors are like, say like, our families are falling or whatever, I guess, like, and then she's underneath 30 degrees and stuff. Yeah. I got doing just fine, but they need the middle name.
I'll find it. Yeah, I would call them.
Can we look at that?
Okay, let's finish this off.
Well, like I said, that was not more.
Oh, wait.
Which one will tell you? That's one that's more your price.
Okay, to kind of refresh our memories, since we lost a lot in the 15 minutes that we were gone, 20 minutes or so. What we're doing here is this. We are following an anabolic process or catabolic process. In this case, a catabolic process, because what are we doing? Degrading glucose, this monosaccharide. And how far have we gotten with this? Well, we've seen where we initially split the glucose. That process was called glycolysis. Was there any oxygen involved in this, or any kind of respiratory gas? No. And this is where students get confused a lot. So this is considered to be then an anaerobic process, but it is not a form of anaerobic respiration. It simply means it's anaerobic because does it require any oxygen here? And your answer is no. In the process of doing that, we had to use a little energy, but we got some back. We got more back than we used up. So it was an energy producing process. We cleaved down this 6-carbon molecule into 2 3-carbon molecules. Those were called pyruvate, right? Or pyruvic acid. Why is there an empty seat next to you? She's gone where?
Wipe them away. No, that's too soon.
Okay. Let's let her know that we missed her.
She's afraid you were gonna make her get up and be a son.
Yeah, that was probably it. So we did this splitting process. One of the other things that happened was that hydrogen was released. And we said that we can't have free hydrogen floating around because it drops the pH. So somebody's got to pick it up. So we had a hydrogen acceptor pick it up. That happened to be something called, did I ask you to memorize the entire name? The answer to that was no. But can you remember NAD? Yes. Guys do. It's easy to remember because we talk about the NADs. Right?
Oh, God, sorry.
So it's an easy thing to remember for us. We've split down here. This didn't require any respiratory gas at all, so it was an anaerobic process, but it is not anaerobic respiration. In our cells, this occurs in the cytoplasm because all the enzymes that are needed for all the steps are there. In bacterial cells, it happens in the cytoplasm because that's where all the enzymes are that allow this to happen. But then we wanted to get as much of the energy back out of this as we could. And there was still a boatload of energy in this 3-carbon molecule. Right? Is there still hydrogen and oxygen here? Yes, we're just not writing them down anymore because I'm trying to keep this simple. But we went through then the process of saying, okay, let's really strip the energy out of this as best we can. And in order to do that, one of the places where that's going to occur is going to be the Krebs cycle. But that's not really the place where a lot of energy gets produced, but it's a processing place, and we call it this party. In order to get into the party, pyruvate had to be reduced to what? An acetyl group. It had to lose what? Carbon and oxygen. There it went. As a 2-carbon compound, it got picked up by coenzyme A, which, by the way, if you go to your Kroger store, you go to your GNC or whatever those goofy stores are, which I hope you don't buy anything in, is that you look there, and you could buy coenzyme A. It says to increase energy output. Bullshit. Has nothing to do with that. You have to have a severe metabolic disorder in order for you to be spending the money on coenzyme A. And none of you in this room do, I would doubt. So it's a waste of your money. So we went over here to the Krebs cycle, and what we did at that particular point was cleave off the other two carbons along with the oxygen. So what got released? Carbon dioxide again. Now, of all of the, and this happens twice, right? Because we did this one and this one too, right? Everybody with me? So now all the carbon is gone. All the oxygen is gone. We actually made a couple little ATP here, but we still have a boatload of energy. And the question is, well, where is the energy at this point? Well, it's bound up with these uncontrollable children called hydrogen. And they're high energy molecule. They're atoms, really, at this point. So we need to deal with them. We've got some over here we still got to dump off. And the question then is, how are we going to get rid of the rest of these? Well, we said that in our cells, where does all this crap happen? On the inner membrane of the mitochondria. And that in bacterial cells, it happens on the cytoplasmic membrane. So what are these other words out here? Well, let's do this first. Let's deal with aerobic respiration, and I can show you real quickly what happens, and it's pretty simple. Here is a membrane. What kind of membrane? Phospholipid membrane. This is inside, this is outside. Now that could be where? Inside of the membrane here or outside, or inside the membrane here or outside. It's on one side of the membrane. And what we have in this membrane, remember what that looks like? I don't have to draw all this. It's that biphospholipid. Remember all that stuff? You do. But what I have on the inside are a series of enzymes. And what these enzymes will do will be to pick up the electron from the hydrogen. And in turn, that's why this chain is referred to as the electron transport chain. Hydrogen is made up of what? One electron, one proton. So the electrons, positive or negative? Negative, are being passed along here. But your question should be, what happened to the proton? Well, the energy that was released from these high energy electrons, we'll keep this very simple. The energy released from these high energy electrons was used to pump the protons out through the membrane. This is called a proton pump. But to use the word channel is right, because that's what it is. It's a proton channel. But in order to get the protons through, which are positive or negative? Positive. To get them through, the energy from passing these hot potato electrons along, that energy released there, and we used to think this is where all the ATP got generated, until we understood chemiosmosis, again, about 20 years ago. Some of you were just born at that point when we discovered this whole process. Now we realize that the energy being released from the electron transport chain, these, and they all have names, I'm not asking you to know them, inside the membrane, passing electrons along, electron transport chain. Chain simply means it's what? A bunch of enzymes lined up together. That's all that means. Where does the energy go? To pump the protons through the membrane to the outside. So now what starts to happen? We start to end up with what? This massive quantity of positive charges to the outside, and what on the inside? Electrons, negative. And these guys want to do what with one another? Exactly. So somewhere else along the membrane, there is another channel, and it's goofy looking. You looked at the electron microscope pictures of these, and it looks like somebody built in their garage. And it looks like a funnel, and at the base of the funnel, it looks like this washing machine that's stuck on the bottom of it. It's the weirdest looking thing. But what it is actually a channel again. But what's going to come through this time? Where did the protons go through this channel? Out. They had to be pumped out by energy. They were out here. Now they're going to come pouring back in. But when they come through the base of this pour-in, this proton pour-in or channel, there is an enzyme at the base, which is an ATP synthase. ACE meaning it's an enzyme. ATP, synthase. Synthase means synthetic, means it makes something. What does it make? ATP. So what occurs? All these protons flooding back through. They spin, virtually it spins this enzyme that looks like a washing machine at the base. It's a unique protein. It spins, it dumps out all this ATP all over the place. But what gets reunited? Proton with electron. Problem. We still have hydrogen.
So why do they?
I'm not done yet. So now we've reunited our hydrogen. What have we removed? A whole bunch of energy from it though. But we still have a problem with hydrogen. So if you are an aerobe, if you undergo aerobic respiration, breathe deep. Okay. That oxygen that you just inhaled now is down in your alveolar sacs. It's crossing the respiratory membrane, which includes what? That simple squamous epithelial cell of the alveoli. There's a slight space in between filled with intracellular fluid into the simple squamous epithelium of the capillary. It's now floating where? By now, it's already back to your heart, going into the left side of your heart, left atrium, left ventricle. Right now, it's going out through your aortic arch. It's now making its way from all the branches off the aortic arch, and it's now by this time even entering arterioles, and from the arterioles, give it within the next 3 to 5 seconds, it's down into capillary beds. And what is the sole reason that you inhale the oxygen? To accept the hydrogen at the end of your chemiosmotic process. That's it. There's nothing magical about oxygen. There's nothing that's mystical about it. There's no kind of vital spirits associated with oxygen. What is the only function of the oxygen you just inhaled? To accept the hydrogen and form metabolic water, which you can either sweat out within the next couple hours or you can urinate out. That process, because who was the final hydrogen acceptor? Oxygen, is called aerobic respiration. Everybody running with me? The process called chemiosmosis, chemi meaning chemicals, osmosis means what? Usually we consider that to be water passing through, and that's where the name gets confusing. It implies what things are going from an area of high concentration to low concentration. Refers to this process whereby we pump hydrogen protons out. The energy to supply the pump came from what? The electron transport chain. The protons came floating back through. This overall process is called chemiosmosis. Because osmosis implies there's a membrane involved. Yep.
Okay, so basically it goes through this entire, goes through the pump and break down and push back through just to get rid of the extra energy so that it can be picked up by the oxygen.
What we're trying to do is milk as much energy out of this as we can. And you raise a good point, because what we left out of this picture was what? ATP synthase. What did we make from this? A whole boatload of ATP. So theoretically, what's the number you always hear? If we started with one molecule of glucose over here, potentially what was the max we can get out of it? 38 ATP. Rarely do you get 38. Usually it's going to, and I'll explain why, but usually it's something around, it could possibly be as high as 38, but usually it's somewhere below that. Well, that's a lot when you think about it. Okay? So what's the scoop of the other two things we talked about here? What's the business with, what was the second option? Anaerobic respiration. Well, I tell you, let's do one thing before we go there. Let's go back up here. Mitochondria, bacterial cell. What do you know about, there's a lot of talk about this these days. What do you know about mitochondria?
They're not because of chloroplasts and bacterial cells. Like they may have derived from bacterial cells.
Okay, but before we even go, and you're on the right track, and that's where I want to go. But basically, what do you know about mitochondria from, what happens every time one of your cells divides? Mitosis. What occurs? We go through all this stuff. Remember the chromosomes shorten up, and they do this, and they all line up together, and then they get pulled. Well, what's going on with the rest of the cell? What happens to all the rest of the organelles? Endoplasmic reticulum, Golgi apparatus. What happens to all that stuff? It's digested, so that the new daughter cells have to make their own. But what doesn't get digested? Mitochondria. In fact, what do they do? When the cell is dividing, what do mitochondria do? They divide as well. Because they have inside them, the mitochondria has this nice single ring of DNA. A single ring of DNA. Sounds like what? Bacteria. In fact, if you compare the plasma membrane on the inside of the mitochondria to the plasma membrane on a typical bacterial cell, you would find that they are identical. Tells you what? What's the theory behind the presence of a mitochondria? Run with it then, John.
It was like a bacteria that we kind of co-opted in this symbiotic.
Use the right word. Endocytosed, phagocytosed, whatever it was. So we brought it in, but didn't kill it.
Oh, okay.
For whatever reason. So what have you got then is this represents, the inside of your mitochondria represents a bacterial cell. It's got its own DNA in the single chromosome in the form of a ring. Bacterial membrane, identical. So what do we got? And what does it do every time your cell divides? It divides as well. Where does this, just getting off the subject for a minute here, why has this been big talk lately? What does it say about your mitochondria? All mitochondria come from the mother, right? So everybody's mitochondria comes from their mother. Your father never donates mitochondria. And then you go, what? But that takes you back then to what happens when that little sperm came along. Your father ejaculated this mass of sperm. They made their way up your fallopian tubes.
I don't have it.
You know, if you have. And in turn, they found an egg, and what did they do when they found the egg? A sperm fused its membrane, and what did it dump inside the egg cell? Only its chromosomes. What got left behind? Its mitochondria. So now all mitochondria are female, right? They're maternal in origin. In fact, the interesting part about this is that if you look at all the potential mitochondria out there, they fall into one of two groups. That's why oftentimes you hear the term, there's Eve 1 and Eve 2. Yeah.
Is that why when they're trying to verify genetic relations of like Egyptian mummies and stuff like that, they go with the mitochondrial DNA?
Lasts longer too.
Yeah, because you can analyze, you can analyze that data because those mitochondrial DNA preserve much better than does regular cellular DNA. The point out of the whole thing, you heard this story last, talking about tribe parents. Did we talk about that in here? About they were raising this stink about now, what do you do with a child who has got a mitochondrial or a genetic disorder, metabolic disorder associated with the mitochondria? What could you do? Is theoretically, if you do in situ fertilization or in vitro fertilization in a dish, you could what? Take a normal mitochondria that doesn't have the DNA problem with it associated with it, and you could insert those normal mitochondria into a cell. So here you've still got a donor father, a donor mother, and we're going to remove the bad mitochondria from the egg cell, and we in turn are going to put in mitochondria from a third parent into that to make sure that the offspring now doesn't have the mitochondrial disorder. But in this particular case, it's really not because that's what it sounds like, but all you're doing is, it's gene therapy, is what it amounts to. In a sense, it's kind of genetic engineering, but it's kind of on the edge of all of that. Okay, so enough of that stuff. So over here then, how do we get to the next group? If you are an anaerobic respirer, you can't have oxygen present. And the question is, well, why not? Well, what do you know about oxygen? Oxygen typically exists in what form? Okay, it's a diatomic type of a molecule. It exists as O2. Why do you take, when you take your multiple vitamins in the morning, you notice that it lists on the back there antioxidants. Antioxidants, vitamin C, what else? Some vitamin B and vitamin E and so on, are antioxidants. What does that mean? Yeah, it means that what oftentimes occurs in very small quantities, but still occurs, is that you end up with radical, highly energized oxygen atoms. And they're called free radicals. And what's the purpose of those kinds of vitamins? Latch on to those because those highly reactive oxygen, even though it's oxygen, you go, this is good stuff. When it interferes with DNA, you have problems, and it sets up what? Cancer type things. So how do we deal with that? We have cells that produce an enzyme called, it goes by a couple of names. Which one should we give you? Let's use this one because it's kind of neat. Superoxide dismutase. Obviously, ACE means what? Enzyme. Mute sounds like what? Mutation, which could be caused by what? Superoxides. And to dismute means to not mutate. So what we have present as aerobes, which bacteria do as well that are aerobes, is that they have the enzyme called superoxide dismutase. And with that superoxide dismutase present, it means that when those superoxides show up, we can latch onto them and pull them out so that we don't cause damage. Which means that we can be in the presence of free oxygen. It can be our final hydrogen acceptor. If you are an anaerobe, a strict anaerobe, and none of us are, but many bacteria are, you lack the enzyme. You're mutated. You do not have superoxide dismutase. Therefore, you cannot be in the presence of free gaseous oxygen because it runs the risk of damaging your DNA. So, how does an anaerobe deal with this? What is it going to say? I can't be in the presence of oxygen, so I have to live in an environment where there is little to none, but I still need ATP and a boatload of it. I need lots of ATP. So what does it do? It goes through all of this stuff we saw before. What is the difference in the process? The final hydrogen acceptor. Hang in there. Which cannot be oxygen. So what could it be? Could be a whole variety of things that may contain oxygen, but it can't be free gaseous oxygen. So it could be things like nitrites, NO2, NO3, carbonates. It could be a whole variety of sulfites, sulfates. Could be a whole variety of other things. And you think about things like sulfite, sulfate, what does that sound like? When you start to think sulfites, sulfur-containing compounds, rotten eggs, the smell of rotten eggs. And oftentimes, where do you get the smell of rotten eggs? If you're ever out in the woods and you go into a mucky pond, which has got nothing growing in it because the oxygen level is down to nothing and it stinks, it's because of the bacteria growing in the mud, which are exuding those sulfur-based compounds. They're living quite well there, and they love it there because there is no oxygen present. Yep.
Is that when it like goes to make COOH? Is that when it makes Q with anaerobic activity?
Carboxyls?
Yeah, like is that when like the CO2 will pick up the hydrogen? Is that what this is?
No, not in this case.
Okay.
Hold on to that. So, what do we have here if it's aerobic respiration? Same process, basically. All the same stuff can go on. There are slight modifications. You don't need to know that. The difference is the final hydrogen acceptor, which cannot be free oxygen, but can be any kind of, she returned, but can be some kind of other molecule which may contain oxygen, but it can't be free gaseous oxygen. Okay, that leaves us with the third option, which is what? Fermentation. And the fermenters are our friends. So what comes to mind when you think fermentation?
Wine.
Yes, okay, so we think right away alcoholic fermentations. And who's best at doing alcoholic fermentations?
People in jail.
Yeasts. Okay.
How does they make it in toilet?
I don't want to know that.
Well, that's how they make it, you know?
Okay, I don't want to know.
Because you've been in jail so many times.
I don't want to know this stuff.
But that's not anaerobic.
Anyway, we think of yeast. Are yeast bacteria?
No.
No, they're fungi, correct? But yet they ferment. We know that some cells in our body will ferment. We talked about lactic acid fermentations before. But there are loads of bacteria that ferment. What are the end results of fermentation? Well, here's where the variation differs. Because what it means is there is no final hydrogen acceptor. Ooh. So do yeasts produce alcohol all the time? The answer to that is no. They'll only do it when what? They get stuck at the bottom of this big thing filled full of grape juice, or stuck inside a loaf of dough where the oxygen runs out. But they would prefer to do what? Be aerobes, but there is no final hydrogen except there is no oxygen present. So then they fall into this mode. What do your skeletal muscle cells want to do? They want to use oxygen. They want to be aerobes. Oxygen's not present, you're forced into fermentation. So what happens if the final hydrogen acceptor is not present? Stops. Stops. What are we left with? Does this involve any respiratory gas? No. So can we still go through glycolysis? And the answer is absolutely. But we still have a problem because what still was produced here? Hydrogen. So how do we get rid of the hydrogen? And the answer to that is this. The final hydrogen acceptor will be pyruvate, or a derivative of pyruvate. And that, depending upon the enzymes that a cell's got, that could be what? We could have pyruvate accept the hydrogen and go through one more, what we call an ethanolic fermentation to produce ethanol, drinking alcohol, which is a 2-carbon compound. And 2-carbon compound, so here comes the hydrogen over here, gets accepted down here. What's the result of this? We end up with a 2-carbon compound, but what also gets given off? CO2. That's the bubbling found in your wine. That's the bubbles found in your beer. Those are the bubbles found in the dough when you make bread. Now, how many other kinds of fermentations are there? That's an ethanolic or an alcoholic fermentation. It's hard for you, I think, to even imagine where you are when, if you would take everything you ate on a daily basis and looked at it, and you'd realize that almost everything that you eat has at least had some form of fermentation associated with it. So let me just give you some examples. Obviously the beer, wine industry, the beverage industry, you know about that. Bread making, you know about that. What about tea or coffee? Coffee gets its unique flavor because the beans are picked when they're still somewhat greenish, and they're allowed to dry in the sun, and in that process there's a bacteria that invades the coffee bean, ferments the coffee to produce this unique flavor so that after it's roasted, it's got this coffee flavor. If they don't ferment, you end up with this something that tastes like dirt or bark. Tea. All your teas are fermented with the exception of green tea. So they're all allowed to be fermented by certain bacteria. So black teas have a certain bacteria that they allow to invade the leaf, to change its particular flavorings and all that kind of stuff. Cheeses, obviously all your cheeses, if they're real cheeses, are the product of some kind of fermentation. Examples of not being that are like American cheese, which is not cheese, right? American cheese is cheese food, which means that we took stuff that we can't even feed animals and we put loads of fat into it and salt, and we feed it to our kids and tell them when they're really young that this is good stuff. Anybody have pizza last night? Okay, what did you have on your pizza?
Sausage and pepperoni.
Sausage and pepperoni. Let's talk pepperoni. Real pepperoni, if it's honest to goodness pepperoni, how is it made? You take, and this goes back thousands of years. You grind up meat and you throw it in some big hole in a rock somewhere or something like that. You add some salt if you've got it, and you add some other spices that you're picking from the bushes around there, and what do? You walk through it squish it all together, and you end up with a bacteria called Pediococcus, which is that same bacteria that causes toe jam. And what it does is gets into the meat, and what do they do then when it's all mixed together? Real pepperoni is hung in a casing, which is intestinal casing. It is not cooked meat if it's real pepperoni, but the crap you buy in the store is all processed. But if you get to a good Italian grocer and you get real pepperoni, it is allowed to hang there. The bacteria then do what? Ferment and produce a whole series of acids, mostly lactic acids, which drop the pH, which means nothing else is going to grow on it. And it changes the whole consistency of the meat so that you end up with this unique flavor and this unique consistency that we call pepperoni. Sauerkraut? Fermented? Yeah, that's all it is. It takes an organism, a bacteria which is found in the air all over the place, which likes to live in a slightly haline or salty kind of an environment, and it ferments the heck out of the sugars found in the cabbage, and you end up with sauerkraut. Olives, all your olives are fermented.
You can't eat ripe olives.
No, you can't. It's impossible. They have to be broken down by the fermentation process and have their whole texture changed. And because then there's so much salt added to those, you got to have a bacterium which is willing to live in that high salty environment at the same time. And we could go on forever. It's tough to find foods that have not been fermented in one form or another. So what makes, for instance, like a blue cheese? What's unique about that, or Roquefort? You know, a good, like Maytag Blue, which is just it's the Cadillac of blue cheeses. Say it again.
They use mold.
They use mold. So what they're using is a bacterium, and at the same time they're using a mold which gives it this bluish, greenish, as the mycelium grows through the cheese itself, and produces this unique kind of a flavor and a consistency to the cheese itself. So what do you do when you see a little mold kind of growing on your bacon or your cheese? You know, scrape it off, or if anything, just eat the stuff.
That's what you do?
Why not?
Because I throw it away.
On your
Maybe I'm
bacon?
just weird.
I've never seen mold on bacon.
Yeah.
Does that actually happen?
If you've got, if bacon is overly processed, it'll stay around for years. But if you buy good bacon, it'll start to mold on you in about 2 to 3 weeks. So anyway, okay, let's finish the story. Who is the final hydrogen acceptor here? Pyruvate, or derivative of pyruvate. Why did we refer to these as, we gave them another name? We called them facultative anaerobes. Facultative anaerobes. If someone is facultative, it implies what?
Cope with a different situation.
Yeah, they can, if it's not this way, I'll go this way, etc. And so what we got is, I wish they wouldn't use the word anaerobe, but that was what has been used for the past 100 years, and so we're stuck with that. Because does it imply that these cells have to be anaerobic? No, it doesn't. It simply means that their respiratory gas of choice is not there. That's all that means out of that. Where would, if you're an anaerobic organism, if you're an anaerobic bacillus, there you are, and you're sitting across the street and in the park across the street in the soil, which is where a majority of gram-positive anaerobic bacterium lie. They lie in the first couple of inches of the soil. What do you know that happens to you on a regular, you think over a 365-day year, what happens to you? Temperature goes up and down, oxygen levels go up and down, water content goes up and down, and so what are you more than likely going to be able to do, because you're always exposed to these horrendous changes in environmental conditions? You're going to by producing endospores. So what we see is that most aerobic bacteria, not all, most aerobic bacteria tend to be spore formers. Okay. That was the whole thing. We did it in an hour and a half. That's everything you ever want to know about respiration. Questions?
Yeah.
Yes.
Ask me.
Did you say the aerobic or
Never ask me
anaerobic?
if I said, because I have no clue.
Okay, so is the anaerobic or the aerobic that tend to be spore?
Anaerobic bacteria tend to be spore formers. Soil, gram-positive, that'll be a next, I guarantee it'll be the next test question, okay? Gram-positive anaerobic bacilli tend to be spore formers, yes or no?
Yes.
Thank you. Anybody misses that, I'll come and get you.
Say it one more time, it's not
Because the fear factor is in there right now. He's gonna show up at my house at 3 in the morning.
Say it again, please.
Gram, who's most likely to produce spores? Gram-positive anaerobic soil bacilli.
That makes sense. Because spores, you need them to survive.
Okay, but then your next question should be, and the answer we will allude to, is that with Clostridium difficile, obviously it's not a soil organism. So where does that play into this? Because it obviously produces spores and it's an anaerobe, but yet it causes disease. And why would there be an anaerobic condition in my colon? So think about that. Think about that. Okay. Can you bear with me for another 15? I think my voice will hold. Any other questions about this? Let's move on to something simpler. Thank God. I'm very proud of you, quite honestly, because having done this for a bunch of years, usually by the first hour, I get people that either leave or go, I don't get any of this. What I would urge you to do is to kind of wander your way through what we yacked about. And the next time we get together, bring those questions back, and we'll go along through all this again. Okay? Anything that you need to have cleared up. By the way,
It's chapter 5.
yeah, thanks for being on top of things. I didn't even know that. So where are we now with your digestive process?
I don't know. I guess, isn't it at that point where it's going?
Exactly. Exactly. So you're still going,
Yeah.
okay.
What's the scientific term for?
I don't even know how to spell it.
Yeah.
Okay, a couple of things then. Because this is going to play into lab that we're going to be doing this afternoon or tomorrow afternoon, depending on when. Oh, by the way, lab, I've got a faculty meeting. This happens like twice a semester, which probably won't be over until 1:30. So regular lab people today is at 1:30 as opposed to 1. Okay? So what kind of factors affect this ability to metabolize? We started to allude to this before. We said you got to have a source of carbon, you have to have a source of energy, right? So we know we've got all that going on, but there are other factors too that kind of determine how well we metabolize. And what are those factors?
How much you're taking in?
That could be a process. I mean, how many nutrients have you got? Do you have the right amount of nutrients? Give me some other things. What other things affect how well you do, how well you feel? Temperature, sure. They're environmental factors. So we got things like temperature. Just let's do these real quickly. pH, exactly. Keep going.
Presence of oxygen.
Okay, let's call it respiratory gases. Good point.
What about water?
Okay, so that would be osmotic conditions. Anything else? Those are some of the big ones. And so what we're going to find is that every organism likes to have certain aside, if all the nutrients are available, let's assume that everything is there that you need to make all the ATP. But these other factors have to be there as well. You know, what happens to you if I stick you in a freezer at home? You know, I decide, I gassed you because you got the answer wrong. And I told you it was going to be on the exam. And you still missed it. And after I repeated it 4 times. And so then I come home at 3 in the morning, I etherize you and I put you in the freezer. Okay, I don't know if I was there. So lo and behold, you're in the freezer. What makes you die? Let's assume that I cracked the door enough that you got enough gas coming in there, oxygen coming in, but what makes you die? Temperature, exactly right. So you fall below a certain body temperature, and what happens is metabolic processes stop. So we've all got what? We've got a certain temperature we like to be at. And our internal temperature is what? 37 degrees C, correct? Okay, everybody knows that. Is there some mystery or mystical number about that? No, it just tends to be an average when you're what? About 20 or 21 years old. It's an average. Okay, does it change over time? Sure it does, big time. When I taught AP, I used to give everybody a thermometer, and for a week you had to monitor your temperature over time. So you had to record, like the first thing you did in the morning before you even got out of bed, you woke up and you had to stick that thermometer under your tongue. You had to lie there for 3 minutes, no movement at all except for get the thermometer in there, record what you got, then record every activity, take your temperature, and so on and so on. You had to do that, students had to do that for a whole week, and their temperatures were all over the place. All over the place. But what happens is that if I can look at that, what would I see? I'd see that there is an average that runs through that, which is about 37 degrees C, 98.6. It's all of this. So there are temperature variations, but every organism, every species likes to have a given temperature at which they metabolize best. And we refer to these then as the cardinal temperatures. So what would these be? Cardinal temperatures. And what does cardinal mean in this case? Don't say redbird. Yeah, so if you're elected to be a cardinal by the Pope,
Your main guy.
yeah, exactly.
It's the main man.
Okay, exactly. So we all have cardinal temperatures, and they go under a variety of categories. There is an optimal cardinal temp, which is that temperature at which you metabolize most adequately and you reproduce most rapidly. So when we're thinking of bacteria, more so than our cells here, what is that thing in the lab? It's called an incubator, correct? And what is that? The temperature of the incubator is kept at 37 degrees because most of the bacteria we use in lab, we got where? From people, exactly right. So the optimum cardinal temperature is that temperature at which the organism can metabolize best and reproduce most rapidly. So if there is an optimum cardinal temperature, there has to be a maximum and there has to be a minimum. And this is where students get confused as well. Because when we're talking about maximum in this case, they always think maximum as it implies best, and it doesn't mean that at all. It simply means maximum in this case is the highest temperature at which this bacterium, a given species can still reproduce. It doesn't kill it, can still reproduce even though very slowly. But if you go 1 degree beyond that, it can't reproduce anymore. It doesn't mean it's going to die. It simply means it can't reproduce. So what would you guess minimum cardinal temperature is? The exact same scenario. It's that lowest temperature at which if you go 1 degree beyond that, you lose the ability to reproduce. So why is fever important?
Keeps bad stuff from
It
reproducing.
doesn't kill, it reduces rate of reproduction. And that's why as soon as you take an organism which has its optimum cardinal temperature at 37 degrees C, and it's causing you an infection, what do you want to do? No, you want to increase body temperature to reduce the rate of reproduction so that your immune system can kick in.
How does that affect reproduction of white blood cells?
It should have minimal effect on them at any point. In fact, advanced temperature will increase activity of leukocytes and lymphocytes.
Which is good.
So, go ahead.
So if your maximum is the highest you can go to reproduce, but still at a slower rate, and your minimum is the lowest,
Yeah.
then when you go past those points, that's when you don't reproduce or you're killing it off.
You don't kill it, you just keep it from reproducing.
Just stop it from
Yeah.
reproducing.
Exactly. So, you know, the whole idea of an Indian sweat hut. You know, the Western Indians used to do that all the time. You got sick, you know, somebody stuck an arrow in you, they broke it off and you came up with a pretty darn good infection. What was the part of the treatment? They put you in a sweat hut. And the reason for that was to elevate your body temperature for a long enough period of time that it did what to those bacteria? It stopped them from reproducing, didn't kill them off, stopped them from reproducing so your own immune system could kick in and kill them off. So fever is a good thing. And yet we've got this idea, you know, God, your kid shows up with a 99-degree fever, you keep them home from school, you know, the kid's got a fever. Send them to school, if anything. Let them raise that temperature even a little bit more. So fever is a good thing. We always want to control stuff, you know, as if there's something mystical and almost godlike to be at 98.6. Fever implies that the body is working, is what it does. You know, you get up beyond a certain point, and of course, obviously, it's dangerous, you know that point. But small, low-grade fever is good stuff, good stuff. So we got temperatures here. Then we can start to use some terms. So what would an organism that is a thermophile? So phile comes from the Greek or the Latin, philios, to love. If you love heat, you would be expected to be found in some place where the temperatures are relatively hot. Where would that be around here?
Not here. And around here?
Water
your
heater.
house?
Could be. Could be. Anybody farm?
All in your manure?
Sure, in a compost heap or a manure pile. You know, you see it steaming, the temperature is 20 degrees below zero, and that thing is just steaming to beat the band because we've got all kinds of thermal activity going on in there. You get bacteria that just love being in that exceptionally high temperature. So we got thermophile. If you're mesophile, meso meaning middle. So you would expect most bugs that like to reproduce at 37 degrees C to be mesophiles. What would you call somebody at the other end maybe? It could be a whole variety of things. You can make one phobic. You could use that term. I mean, the point is you can come up with a whole variety of terms out of all of that. pH. Most organisms like to live about what?
7.
Yeah, about 7. They don't like to live really at 7 because what happens at 7? Nothing, right? 7 means nothing's going on chemically, correct?
Right.
Because everything's copacetic. So they like to either live just a couple of points above 7 or a couple of points below. And yet, you know, what do you find at home? Let's see if I can come up with an example. So let's see what you can come up with. So we got some bacteria that like to live where it's very acidic, and they're called acidophiles. Where might you find an acidophile?
In your stomach.
And that's a great guess. And most people usually say, what about Helicobacter pylori? The bacteria that causes what? Ulcers. We know that ulcers are caused now by bacterial infection. The whole world knows that. And then you would say, boy, we got this bacteria, Helicobacter, don't look at that clock, Helicobacter pylori, and you say it's living in the stomach. Well, it must be an acidophile, but it's anything but. Well, you go, how does that make sense?
Oh,
Well,
is that why it causes ulcer because trying to get out?
what? Say that again.
Is that why it causes the ulcer because it tries to get out? You know, like,
No. No, what it does is, here's your stomach. That's your esophagus there. Duodenum there. And let's say that your ulcer occurs here. If we would look at that, what we would see is that here's the lining of the stomach with its gastric pits and all of that kind of stuff. And these bacteria are living quite well here, eating away at this mucous membrane. And yet they have this mucoidal sheath that they've secreted around them, which is a buffer, a very strong buffer. They can't afford to live in an acidic environment, so what do? They modify the acidic environment to be able to live there, but they're not acidophiles. Give me another example. Where would you find an acidophile? You can't come back here until you show me that you found one. Go home, find one.
In your fridge.
Look at yogurt.
Yogurt, good example. Orange juice, another good example. Great example.
Coca-Cola.
Yeah, because you'll get, if you get a fair amount of backwash, if you're one of those who opens the 2-liter bottle and takes a big swig out when nobody's looking and throws it back in there. And then, and even with milk, it's even better with milk actually, because milk is naturally acidic, is it not? Lactic acid, it's got all that in there. Any kind of dairy product is going to have that stuff in there, but you may end up with some growth on there. Certain cheeses, absolutely. What about vinegar?
Pickles.
Okay. Certain kinds of pickles, yeah, it depends on the kind of pickle that they happen to be. If it's a brine pickle or if it's a... Okay. Yeah, it depends on how the pickle is made. Say what?
Cleaning product?
Probably not. Probably not, yeah. Most are strong bases, they're contain ammonia or alkaline compounds. Anyway, but like vinegar turns cloudy after a while, and it's bacterized. That's what's going on, so. So pitch it out of
You're
that.
talking about like straight white vinegar? Sorry.
It'll happen with any vinegar. Any vinegar, because
Cloudy.
what is vinegar? Yes.
I don't have the answer, but I have a question.
Well, which would be the question or the answer? Let's hope the question's good. We'll go with that first.
Well, my mom uses like vinegar and water to clean stuff.
Sure.
So why is, like, what's it, like, why would someone use vinegar and water to clean it when that's really acidic, but then most cleaning products are really basic?
It's a good question.
It's more about chemical and natural,
The
right?
vinegar is a good cleaning product. It's been used for millennia as a cleaning product, and it just works well. Is it been any better than others? I'm not claiming that. But a lot of people use it for everything from washing windows to cleaning floors to cleaning their dentures. So, yeah.
To go back to one of our lectures from nursing, we were talking about, STDs and things,
Mhm.
and they were talking about you can use yogurt to help with certain types of things going on in a woman's.
Yeah,
But
it's used as a douche, but in that particular case, what they're trying to do is refluorate.
Okay, so
They're trying to-
wouldn't a woman's vagina then be kind of acidic at certain points?
It should be acidic. And it normally is acidic. And yet it's got its own flora, but it's an acidic environment.
Okay.
Yeah, and should be so. When it loses that acidity is when you're going to end up with problems like yeast infections.
Yeah.
Yes?
Isn't vinegar like the product of alcohol and non-alcohol?
You're getting close. You're getting really close. Can you run with that and tell me why?
The fermentation they use yeast, and I don't know, my boyfriend explained to me he's an alcoholic, so I like forget now.
That was nice to share that. So yeah, what happens with a vinegar is that it's what? Acetic acid, correct? It's just acetic acid. And you know, how do you end up with red wine vinegar or apple cider vinegar or white vinegar or whatever vinegar it happens to be? You can flavor them any way you want to. But what you've done with developing those is that you've gone through an ethanolic fermentation first. So you went through that whole process of taking C6H12O6, you went through glycolysis, you split it down here. What happened was pyruvic acid became the hydrogen acceptor, and you produced what? ETOH, ethanol. And then what you do is allow a bacterium, which is what we refer to as an acetobacter, which then uses the ETOH, this 2-carbon compound, as an energy source, and it produces then an acetic acid, so it converts ethanol into acetic acid. So the acetic acid is the vinegar. So you can get a great quality red wine vinegar if it's made with good wine. If you buy like off the shelf red wine vinegar at Kroger's, most of the stuff you're going to get is crap, and you can't even drink the stuff or make even a dressing from it because it's got this off flavor to it, because they've used cheap wine that no one would drink as the source of it. But you're right on the money. So it's the after process of an ethanolic fermentation. Okay, almost at the end here. Respiratory gas, we went through that, right? So we know all those kinds of things, what stuff's got to be there or not present there. Osmotic pressures. How usually do we change osmotic pressures? Not really so much by changing the water, but changing what's dissolved in the water, the electrolytes, typically, more so than anything else. And what's the most common electrolyte that we add? Sodium. So sodium chloride and all of that. So what is an organism that's this? The word is halophile, H-A-L-O-P-H-I-L-E. P-H-I-L-E means to love something.
Like halogens?
Yes, and where do you find halogens on your, you don't have a periodic table in your periodic chart. Where do you find them? If you were to look just there, if you're looking at the thing, right or left? Right. Right side. Halogens. What falls into the halogen group? Chlorine, borine, fluorine, all those things, because halogens react with metals to form chemistry 1, lecture number 1. Salts, exactly right. Exactly right. Salts. So if you're a halophile, you are a salt lover.
I'm a halophile.
So do we find certain organisms which are halophilic? Well, sure, but they're kind of in goofball places, like the Dead Sea, or the Salton Sea, or the, what's out there? Mormon land? Salt Lake. Okay. So we got all that kind of stuff going on. Are there bacteria that live there and live quite well? Absolutely, absolutely. But how have we used the fact that there are so few halogens, halophilic organisms around? Because of that, what do we do with salt? We use it as a preservative. Exactly. Because it creates such osmotic pressures that in many cases it will just kill off the bacterium involved. So what's the scoop? Last item up for bids today. What's the scoop on something that we call a red herring? The term is used a lot. A red herring. Bill Clinton. Remember Bill?
Pickle herrings.
I did not have sex with that woman.
Sexual relations.
Whatever. Herring. He used the term red herring a lot, and yet you'll hear it a lot as well. What does that mean? Any idea? When herring is a fish, exactly right.
Something you used to throw off dogs in a hunt.
No, I don't know where you got that one. That's a great answer. Anybody have a clue?
It's a distraction, isn't it?
A red herring refers to somebody gave you a deal that stank. So if I slip you a red herring saying, I've got a great way for you to invest your money, and this is a non-loser, and I know this guy in New York who's got this scheme, and you can't lose on this Ponzi scheme. You can't lose, okay? So, and where the name comes from, it obviously has something to do with fish. And where it originally comes from, and I think it's in your author actually talks about it if you ever looked at it, is that how do you use to ship fish?
You have to
Salt, right? Because if you're taking cod off of Maine and you want to get it to St. Louis in the 1800s, it's going to take weeks, maybe months to get there, and it's going to rot before it gets there. So what do? They packed it in rock salt, and they still you still buy salt cod, although we don't have American cod anymore because it's overfished. That's a whole nother story. But so let's say that you get this shipment of cod. You own a restaurant in St. Louis, and you ordered this very expensive supply of cod. It comes in, and you rip open the box there, and you scrape away the rock salt, and something smells bad. And so you dig down through this, and the first fish, they're dried like a piece of wood, and you come down, and all of a sudden you've got this fish which is red. And it's the one that stinks. And it stinks because two things. One, there was a halophilic bacterium that grew quite well on it. And the halophilic bacterium typically produce a red pigment. So if somebody slipped you a red herring, it meant you got something was bad out of the deal. So if there's one thing you remember out of this course, aside from the fact that don't trust anybody, it ought to be now what the heck a red herring is. Okay, I've talked enough.
You're
Honestly, this is to you guys for hanging in there. This is a lot of stuff.
I gotta work at
$130.